And I'm John Dinman, and since it's Science on Tap, man, we can't really see that. Imbibe. So I'm going to share my screen here, and we'll get started. All right. So just a pretty picture of our campus. It's a really gorgeous place, and I put this is a stock photo, which you can find online at UMD, but I put this up just to show you that, yeah, there's my office, and I'm really working. So I'm going to be talking about targeting program -1 ribosomal frameshifting in COVID-19. And before I get started, I just want to acknowledge the people who really do the work. This is the thesis project, PhD thesis project of Jamie Kelly. She's a third-year student in the molecular and cell biology program, and specifically our virology subprogram. Alex Olson, she's actually in the Department of Chemistry and Biochemistry. And then Kevin Tu is a Banneker Key Scholar, undergraduate, smart, nice guy, and he's really helped a lot. My collaborators are Mike Woodside at University of Alberta, Kylie Ken Hall at Virginia Tech, she does the virology, Lois Pollack at Cornell, and Jay Schneekloth at the NCI. And the money comes from the NIH, the Defense Threat Reduction Agency, which is basically the Defense Department's version of the NIH. And then, as Abby mentioned, this COVID-19 UMD seed grant. So, okay, how come there we go. So just an outline of my talk. I'm going to talk about coronaviruses and COVID-19 and the virus that causes it, which is called SARS-CoV-2. And then I'm going to segue into this molecular mechanism called ribosomal frameshifting. It's a critical switch in the developmental program of all coronaviruses. And then really going to talk about how we can use it, how exploit it as a novel target for antiviral therapeutics, and then finish up on some of the work that we're doing right now. So this is the picture that you see on every news channel and every TV screen whenever they're talking about COVID-19. But I think this was the best Halloween costume ever. This is so cute. So let's just get over some terminology. So SARS-CoV-2 is the virus. It was previously called 2019 nCoV, the disease is called COVID-19 or coronavirus disease 2019. And this has caused a pandemic. And a pandemic is worldwide or spread over a very large area of an infection that crosses international boundaries and usually affects a large number of people. And I think that we can safely say that's what's happening with COVID right now. And clinically, what happens is, so this is sort of from this study down here. And actually, let me turn on my pointer. Nope. Sorry. Laser pointer. All right, there we go. So, whoops, I went ahead. So these were symptoms that came out of this paper out of China very early on. And this #1 symptom up top is basically loss of smell and taste. And that's exactly what happened to me. Mid-May, the trees were in pollen and there was tree pollen all over the place. And on a Wednesday, I got a runny nose and I figured, it's just, seasonal allergies. And that kept up until Saturday night. We ordered out from a local Chinese place. We got this really, really hot dish. And everybody in my family was like, and I was like, it's not hot. I'm not having a problem. And I realized, oh crap, I really got COVID, And so the next day is when I spiked a fever. I was home for about a week, just could not get rid of the fever. And then a lot of cough, huge amount of sputum production. It was really, really disgusting and difficult. And I just could not break the fever. I had really sore throat, headache, chills, myalgia, which is muscle pain. And after about a week, I just crashed, got shortness of breath. And so I went to my wife and I said, it's time for me to go to the hospital, check me in the hospital. And I was there for 8 days. Fortunately, I was the first patient in Montgomery County who got this experimental drug remdesivir. And the reason I got it is because I told my doctor, I'm a virologist, I want this drug. And they had to go through a whole institutional review board meeting and stuff like that to get it to me. And it really helped. And thankfully, I am absolutely symptom-free. I have no lingering long-term side effects. I think it was probably because I was in pretty good health to begin with. But I will say my father-in-law succumbed to COVID-19 early in March. The rest of my family got it. My wife got the muscle pain and diarrhea and that side of it, which is also not very fun. My kids, on the other hand, one had a fever for a day and then was sort of tired for a week, and the other one just lost her sense of taste and smell for about 4 days, and that was it. So it tends to affect the elderly, like me, people over 60, much more severely than young people. But that is not to say that young people don't succumb to this virus either. So coronaviruses are not new. We've had these 4 varieties for flavors of coronavirus circulating in the human population for thousands of years. They've co-evolved with us over thousands of years, and they cause about 25% of what we call common colds. The other 75% are mostly rhinoviruses or adenoviruses. But more recently, we've seen the emergence of these novel coronaviruses. So this started with SARS, or severe acute respiratory syndrome coronavirus, in 2013. And we narrowly escaped a worldwide pandemic. We only escaped it because of good government and good international cooperation. In 2012, we sort of got another version of a coronavirus. It also caused respiratory syndrome. And that came out of the Middle East, from camels, actually. And then more recently, at the very end of 2019, we saw SARS-2 come online. So these viruses, they're called zoonoses. And a zoonosis is basically zoo, which is animals, and osis is a disease, right, or a condition. And with these coronaviruses in particular, bats carry them. Bats have one of the cool things that come out of COVID-19 research is deep investigations into bat immunology. And it turns out that bats have amazing immune systems. And then what happens is, bats poop and pee, and there's animals on the ground that pick up that and they get infected with it. And then what has happened is that live animal traders, or people, they capture these animals, they bring them to live markets, or they get the camels, they herd them and bring them to live markets. And now you have all these animals tightly packed with one another, typically caged, and again, pooping and peeing on one another, and and they'll be slaughtered, right? So now you've got blood added to that. And it's the workers who are working with these animals. They're exposed, they have multiple exposures every day to this virus, right? In the feces and the urine and the blood. And it's just a numbers game. If you have enough virus and enough times you encounter that virus, eventually a mutant is going to arise that infects the human. And then you start passaging in the human, and you're going to have a mutant that arises, which is able to pass from one human to another. And typically what we've seen with coronaviruses, people get sick, they give it to their family, and they go to the hospital, and then they get healthcare personnel sick, and then the healthcare personnel get their families sick, etc. We also see what's called nosocomial transmission to other patients in the hospital. And this is why, since this is airborne, this is why, for example, my hospital room was under what's called negative pressure. And what that means basically is they vented all the air out of my room into the outside, and made sure that air was not recirculating into the hospital to infect other people. So with COVID-19, well, we start in Wuhan. It's a big city. It's a medium-sized city by China, but 11 million people. And it was started out in this live wholesale market. So you have wild-caught animals, which are being brought to market. They're being kept there. The people who are working with them are slaughtering them on the site, etc. And this then establishes the transmission. Now, unfortunately, this market is right next to the train station. And so it spreads from the train station, spread through China. The Chinese government really clamped down and managed to control it eventually. But really with COVID-19, what happened, it spread we got two different routes of transmit or infection. One was to Italy, and it got to Milan, and I think this corresponded with Fashion Week in Milan. And so, Chinese buyers and suppliers go to Fashion Week to check out the newest fashions and sell their cloth and that kind of stuff. And then a bunch of them took a field trip to Venice, because when you're in Italy, you go to Venice. And so that's how we saw this spread through northern Italy. And then when everybody began to freak out, Americans left Italy and in Europe and brought it to the East Coast of the United States for the most part. And it became established in ports of entry like New York and Boston and the DC area. I mean, we have 3 international airports, and that's why it was so bad here in March, April, May, June. The other route was through the West Coast, and ports of entry there: Los Angeles, San Francisco, Seattle. And this is where we saw big outbreaks early on. So it really, this globalization has enabled viruses and other infectious agents to spread across, all the way around the world within a day. And we saw this with, remember Zika, remember West Nile, the same thing. So globalization is a real driver of epidemics now. These are stats from today. So currently there are 50 million plus cases in the world, and over 1 million people have succumbed to COVID-19. In the United States, we're at 9 million plus, almost 10 million confirmed cases, and 235,000 plus deaths. And we're now in the 3rd wave of this infection, and we're experiencing over 1,000 deaths a day. This is, as Joe Biden said, this is going to be a very, very dark winter. So be careful. So this is a coronavirus, and the way coronaviruses get their names is what they look like when you take a picture under an electron microscope. And so what they see were these bumps on the outside, and it looks like the Burger King crown. So they called it a corona for crown, based on its crown-like appearance. And so what we've got is these spikes on the end, on the outside. These are called the spike proteins. And this is what binds to that ACE2 receptor to get into on the surface of cells to get in. And then it's called what we call a envelope. So this is a lipid bilayer. It's basically a membrane. It's derived from the membranes of the cells that it affects. And then on the inside is the nucleic acid genome. Unlike our cells, where we have double-stranded DNA as our genome, this virus has a single-stranded RNA as its genome. So it's really interesting that viruses, they don't need a DNA genome like us. They can use RNA as well. And this RNA genome is wrapped up inside of this N protein or nucleocapsid protein to sort of protect it. So that's basically what the virus looks like. So if we look at its genome, it's got a big genome for a virus. Its genome is about 30,000 bases long and single-strand RNA, as I said. And for most RNA viruses, genome size is about 10,000 bases or even less. So this is a really big, honking big virus. And when you talk about the molecular biology or the genes, it's basically, these show the genes. So you have a gene called ORF1A and a second gene called ORF1B. And then you have a bunch of genes downstream called ORF2 through 9. ORF means open reading frame. It's just our jargon. So what happens is when this virus infects cells, it dumps its genome into the insides of the cell or the cytoplasm of the cell, and the cell's protein synthetic machinery called ribosomes, they hop on this end or the 5' end, and they start to read the genetic code and make the proteins that encode non-structural protein or NSP1 through 11. And the function of these proteins or the non-structural proteins is they hijack the host cell. They interfere with the ability of the host cell to recognize that it's been infected with a virus and to send out the alarm. So it's like, in a movie chase scene, the detective is chasing the bad guy, and they go down an alley, and the bad guy starts throwing garbage cans and whatnot behind him to slow down the detective and maybe escape. And that's what these non-structural proteins do. They're slowing down the host immune response. And then in this region where open reading frame 1 and 2 overlap, is this signal called the program -1 ribosomal frameshift signal. And what it does is it directs a fraction of ribosomes to skip out of this frame and skip into this one and now translate this ORF1B open reading frame. And this encodes non-structural proteins 12 through 16. And the function of these proteins is to make RNAs. So it makes new genomic RNAs and subgenomic RNAs. The subgenomic RNAs come from here. And these encode structural proteins, which are used to basically now take the new RNA and the structural proteins and make new virus particles. So if we diagram this as if we're computer scientists, we basically have a program first what we do is we infect the cell that initiates the program. And then we have this process of making ORF1A proteins, and their function is to hijack the cell. And then we get to this decision point. Do we frame shift or not? And if we don't frame shift, we just go back and keep hijacking the cell. But if we frame shift, and this happens only a fraction of the time, then we make the proteins to go to step 2 of this program to synthesize RNAs. And once the RNAs are synthesized, we can make the structural proteins, we can make the new genomic RNAs, and new viral particles can assemble inside the cell and then exit to infect the next cell. So this is a computer program, and a very simple program. And what's really important is this program ribosomal frameshifting is this really critical nexus point. If frameshifting doesn't happen, we stay in this phase. Frameshifting does happen, we initiate the second phase. So this is a really critical nexus point for the viral developmental program and something we should be able to target. So now I want to talk about sort of how frameshifting happens. So the frameshift signal is a bunch of elements, a few elements on an RNA molecule. And they're basically 3. We have what we call a slippery site where the frameshift happens, a little spacer of maybe a dozen nucleotides, 2 to a dozen nucleotides. And then this RNA structure called an H-type pseudoknot. And what happens is that an elongating ribosome comes chugging down the messenger RNA, and it hits the pseudoknot. This is like a speed bump, and the ribosome has to stop. Well, and while it stopped trying to unwind this structure, the tRNAs, the transfer RNAs on the ribosome, are base paired to this NNWWH, the sequence. And if there's a shift by one base backward or in the -1 direction, the tRNAs can repair what we call their non-wobble bases to the -1 frame codons, or NNNWWW. And eventually, the ribosome can resolve the structure and continue translating now in the new ORF1B, or the new reading frame. So one of the questions we had many years ago are, why are these pseudoknots stimulate frame shifting much more efficiently than stem loops? So this is a stem loop. It's basically from here to here, we have a little stem, a loop, and we come back on the other side. The pseudoknot is a stem loop structure where sequence downstream can come back and base pair to this loop. And if we look at frame shift, or like the percent of the 0 frames, so here's a 0 frame control, that's 100%. If we look at something that contains a pseudoknot, we get about an order of magnitude less translation through. If we have just the stem loop, we have an order of magnitude less than that. So these pseudoknots really promote efficient frame shifting. So to explain this, we came up with what we call the torsional restraint model. Essentially, as a ribosome elongates into a stem loop, there's some resistance to the forward motion, but basically the distribution of the unwinding energy is distributed pretty equally along the length of this sequence. But if you have a pseudoknot, what happens is you unwind this side, or the stem 1, this side gets all knotted up, it gets supercoils on itself. And basically the ribosome can't go through the structure until this part has been totally broken. And so what this does is direct or focus the unwinding energy at a point where the forward energy is counteracted by the backward energy, or the resistance to this caused by this supercoiling. And that then directs the ribosome to pause at a very specific site. And if your spacer is the right region, it directs it to pause it with the tRNAs over the slippery site. So that was an idea. I want to give you an idea of how science proceeds. That was an idea that I got back in 1993. And you can see that the paper was published in 2005. It took me 12 years to come up with the experiment to figure it out. And that's how scientists are. We're like a dog with a bone. We will gnaw at it forever, a question forever, until we finally maybe get the inspiration to figure out how to test it. So the experimental design we came up with was called a pseudo-pseudoknot. So we start out with a reporter with just this simple stem loop, and then we take a synthetic piece of DNA and we base pair it, we hybridize it, so we allow the two to come together, so we can make what we call a false pseudoknot, or a pseudo-pseudoknot. One side of the piece of DNA binds here, the other side binds here, and then we can add 1, 2, 3, 4 bases in between, just to sort of give it more or less play. And when we did, so the idea is that these extra bases are added to give it more or less play, and we can recreate the pseudoknot with this false pseudoknot. And here are the data. So here's our 0 frame control. This is where we have the joining oligo just has one nucleotide, two nucleotides, 3, 4. And we see that we can really sort of fine tune it to optimize frame shifting efficiency, and really 40% frame shifting efficiency is very high. So now we understand how frame shifting works. Let's see if we can, if it's a target for antiviral intervention. So to back up here, frame shifting was actually discovered in HIV in 1985 by Harold Varmus and a very talented postdoc in his lab named Tyler Jacks. Now, Harold Varmus, he's won the Nobel Prize for discovering reverse transcriptase. And I was very relieved when Harold Varmus was named the director of the NIH, and he decided to not study this because I couldn't compete with him. And Tyler Jacks just went on to a faculty position at Harvard where he got interested in cancer. So good for me that they didn't continue studying this, because they're really smart. So we see in HIV this overlap between this gene called GAG and PAL, where PAL's in the -1 frame with regard to GAG. And this is where the frame shift was discovered. Now in coronaviruses, about 5 years later, a good friend of mine named Ian Brierley at Cambridge used a coronavirus to discover the pseudoknot that promotes frame shifting, -1 frame shifting. And then I had started out working with this virus called LA. It's a simple virus of yeast. Yes, yeast have viruses. In fact, all of our brewing strains, here we go, have this LA virus in it. And back in 1989, we had just gotten the sequence and we saw that this GAG-PAL overlap. And it was obvious now to us that there was a frame shift event. So we have this viral messenger RNA, the frame shift signal in between. And if you don't frame shift, you make, pardon me, you make the, only make this proteins which are encoded by the 5' open reading frames. And if you do frame shift, you make this fusion protein, this GAG-PAL fusion protein. So I was working with this system called the yeast killer virus system. It's really fascinating because you have a double-stranded RNA, virus with a double-stranded RNA genome, which is called LA, just because it's a large genome, and it's encapsulated in these viral particles. And its messenger RNA gets translated in this frame shift. Without a frame shift, you make the blue proteins, which are GAG or the capsid proteins, right? This is the package for the virus. And then when you frame shift, you make the PAL, which is this red thing. So you make this GAG-PAL fusion protein. And then these come together to make these viral particles, and they encapsulate new plus strand RNA. And you can see that you need a lot of structural GAG proteins, the blue ones, and just a little bit of the red one to replicate the viral RNA. Now, this is one of the cool things about virology is you have viruses of parasites, and then viruses have parasites of their own. So there's a parasite virus, or it's called a satellite virus called M, because it's got a mid-sized genome. So it's got its mid-sized genome, which is about half the size of LA, and it steals GAG and PAL, the proteins from LA, to encapsulate and replicate itself inside of viral particles. And interestingly, M makes a protein called the preprotoxin, which is then cleaved by these proteases called KEX1 and KEX2 for killer expression, which then makes the cells that are infected with the virus secrete a toxin. And so here's cells that are infected with the virus, here's cells that aren't, and these have been put on top of a lawn of cells that aren't infected with the virus. And you can see that these cells over here can't grow into this zone. It's because the virally infected cells are secreting a toxin to which they're immune. And interestingly, this KEX1 and KEX2 turned out to be the furin proteases, which are involved in, have lots of medical applications. And this is an example of how, basic, simple yeast virology has identified things that are really important for medicine. So I was a postdoc at the time, and I set up a reporter assay. Basically, ribosomes start here. They translate through this sequence that we put into a gene called LAC-Z, which encodes a protein called beta-galactosidase, which if you put the right stuff into the media, it'll turn it yellow or blue or any other color you want. And the bluer it is, the more color, the more frame shifting. So these are just controls that don't have any insert in the middle. And then these are the tests that do have the insert. And the important thing is this one, where with the insert, with the viral sequence, and LAC-Z is in the -1 frame, we got 1.8% frame shifting. So we were able to measure the rate at which frame shifting happens. And I happened, as a postdoc, I remembered my virology class. I knew that this was a simple, what we call icosahedral virus. So it looks sort of like a soccer ball. And if you count the faces on a regular soccer ball, a regulation soccer ball, you'll get 60 different faces. And indeed, these viruses basically are units of 60, 60 to put together this viral particle. And 1.8% of 60 is approximately equal to 1. So we got this idea that maybe this virus has fine-tuned its frame shifting to deliver 1 copy of this for 60 copies of that, right? And if we upset it, what happens? So working in yeast, we have some really nice molecular genetics available to us at the time. And we were able to manipulate the system so we could increase or decrease frame shifting. And when we increased or decreased it, we see that M totally disappears. And this zone of growth inhibition, this killer assay, we can see that the killer zone becomes smaller and smaller until it disappears. So this suggested that frame shifting is efficiency frame shifting efficiency is critical for viral propagation. And the idea is, it's a supply side thing. Normal frame shifting efficiency gives you the right ratio of structural to enzymatic proteins to replicate your virus. If you have too many or too little, the process of viral particle assembly crashes. And the idea, for example, let's say you're making cars and you see you need 4 tires for every chassis. And you set up your supply chain to make sure that that's the right ratio. And then you hire your idiot son-in-law who hires three tires per chassis or 5 tires per chassis. And eventually, you're not operating at peak efficiency. You lose money and you go out of business. So if this is really important, then frame shifting should be druggable. And I really don't want to go into the details about why we chose these 2 drugs, but we did. And we found that this one, anisomycin, decreases frame shifting, both in yeast and in vitro. And this one, sparsamycin, stimulated frame shifting both in yeast and in a test tube. And when we grew yeast in the presence of these drugs, we saw that the killer virus rates of cells that had maintained the killer virus went down. And in this, what we call a northern blot, we see we were able to completely cure the yeast of both the LA and M viruses. And just as somebody who works with these viruses, it's really hard to get rid of. So this was suggested that this is a viable approach. However, the drugs that we did use are really, really toxic and would kill a patient. So these were sort of off the table. Once we reported that, another person, his name was Simon Green, and he worked for a company called Ribosomal. In the late 90s, he did a drug screen and identified this compound, which we'll call 4-HCI, which, RG501, and he identified it as something that stimulates frame shifting from HIV. So we have higher frame shifting in vitro, higher frame shifting in cell line, and then what he treated cells that with the drug and then infected with HIV, and we see it really knocked down HIV levels at doses that were non-toxic. It also inhibited decreased viral production from cells that were chronically infected. So this was really promising until the company went out of business, and really nobody's doing this anymore, which is really a shame. Now we started working with Kylene Ken Hall, who's now at Virginia Tech, and she works with these really nasty viruses called arboviruses. They're in the family alpha virus. And these things, there's Venezuelan equine encephalitis virus, Eastern and Western equine encephalitis virus. So the Venezuelan is South American, although it's now spread up into Florida. And then Eastern and Western are indigenous to North America. And Eastern has become a really valid threat. More recently, the CDC put out a warning last summer about it. Typically what happens, it circulates among moose. So occasionally, 2, 3, 4 times a year, a moose hunter, either in New England or Upper Peninsula of Michigan, will kill a moose, butcher it, get themselves infected and die. And it's a horrible way to die because it causes encephalitis. So basically it blows up your brain. Now, we're interested in it because it was weaponized by ourselves and the Soviet Union during the Cold War. It's transmitted by mosquitoes, and these mosquitoes are moving north with climate change. And it basically, there's no approved vaccines or therapeutics. And so this is a map of its genome. It has these 4 non-structural proteins up front. And then once this is the protein that makes the RNA, makes RNAs, makes the subgenomic RNAs that makes the structural proteins. And here at the end of what we call the 6K protein is this frameshift signal. So the frameshift signal looks like U, UUA. We were able to introduce silent mutations that code for the same protein, but mess up the slippery site. So G, UUC, UUG. And so no difference in protein sequence, but the slippery site is messed up. And when we do that, we see that frame shifting goes down below 1 from about 3.5%. And these are additional controls. So we were able to really knock down frame shifting about tenfold. And then we took this mutant frameshift signal and we put it in a real live virus or infectious clone of virus. So this is called, the infectious clone is called TRD for Trinidad donkey, because it was isolated from a donkey that died in Trinidad. This is TC83, which is what we'll call it. This is a vaccine strain, but it's a really crappy vaccine. It's given to people who work directly with the virus. And I've talked to people who've gotten it, and they talk about the TC83 headache. It gives you a massive headache for about 5 days. So nobody wants to use it. And so what we did is we took mutants with the frame, we mutated the frameshift signal, and we infected with 10 to the 3 or 10 to the 4 infectious particles, or the Trinidad donkey, or with higher or lower doses. And what we see is basically, we then gave it to mice, and it killed, the wild type killed the mice. But when we knock out or inactivate the frameshift signal, what we get is either 90% of the mice survived at the low dose, 70% survived at the high dose. And when we looked at the mice that survived and we looked at their antibodies, they had really high titers of neutralizing antibodies. And then we took these mice that survived and we reinfected them with this Trinidad donkey at the high dose, 10 to the 4 dose. So that's the one that killed all the mice within 8 days. And we see that those who got the vehicle or the mock, they died very quickly, but all of our mice that got the frameshift, the mutants, they all survived. So this suggests that frameshifting mutants, we can make mutant viruses and use them as live attenuated vaccines. So this is another strategy that can be approached. So we have drugs and we have vaccines that we can make. And this is just sort of looking at the pathology. The mutants basically, they accumulated more slowly in the blood. They spent more time in the spleen, which is probably why we got this enhanced immune response. And then they were much slower to move to the brain and got cleared out very quickly. So now I want to segue to SARS-CoV. And this is the original SARS. So when we looked at it back in 2005, we saw this interesting structure where we have the pseudoknot. So this is slippery site. Then we go to stem 1 of the pseudoknot and stem 2 of the pseudoknot. But we have this internal stem loop, which we call stem 3, and that was novel. Nobody had ever seen this before. So we made mutants. One mutant, which is all U's of the slippery site, increased frame shifting, and this mutant, U, and followed by 6 A's, decreased frame shifting. And again, we made infectious virus and then infected that into cells. And we saw that both mutants, the high frame shifter and the low frame shifter, knocked down viral titers. So we went from, let's say, this is genomic RNAs and subgenomic RNAs. We went from, let's say, 4,000 copies per cell to 4 copies per cell. So we're seeing a 3 order of magnitude or 1,000-fold difference in the amount of virus produced just by mutating the slippery sites. And then we also made these 3 mutants. These are silent for protein coding. And so we disrupt stem 2, and we see basically the same thing again, where we knock down viral titers significantly. So this suggests that by altering frame shifting, either by changing the slippery site or by changing the structure that promotes frame shifting, we can make attenuated viruses that don't grow very well. So that was back in 2005. We got, the government was very interested, NIH was very interested in learning more about SARS-CoV. So we got a really nice grant from the National Institute of Allergy and Infectious Diseases. And we studied this thing for 5 years. And then when it came time to renew the grant, well, it was 2010. And I was told by the program officer, we're not interested in coronaviruses. It's never going to happen again. So we didn't get funding. And I think that was really, not a smart decision on the part of the NIH. And unfortunately, I have to say, they're sort of driven by the crisis of the moment. And it's not, I don't think it's the smartest way to go. So then, of course, SARS-CoV-2 comes out with COVID-19, and suddenly everybody's interested. And so we look at it again, we have this open reading frame 1 and 2, they overlap. And we have a standard slippery site, the 3-stem pseudoknot. And there's this little bit upstream called an attenuator hairpin, which basically is a sort of push you, pull you, it actually knocks down frame shifting a bit. And when we align the frame shift signal from the original SARS-CoV with SARS-CoV-2, there's only a single difference, the C to A change. And it's in this loop where things don't make much of a difference. Looking at the attenuator hairpin, there's more changes upstream. And initially we thought it might not be functional. So anyways, we did some structural biology work with Lois Pollock at Cornell, and she used this thing called small angle X-ray scattering and compared the profiles of SARS-CoV and SARS-CoV-2 frame shift signals, and she saw that they totally overlap. So this told us that the structures were identical, but interestingly, in this area right here, you see there's a lot of variability. And this is she tells me this is the sweet spot in Sachs analysis, telling us that there are multiple conformers, multiple different shapes. So this thing isn't just one shape. It's really assuming a lot of different shapes. It's sort of like a Rubik's Cube, and it's flipping from one shape to another. I think that's important for frame shifting. So more recently, let's see, my colleague and collaborator, Michael Woodside, he has been doing some what we call molecular dynamics simulations. So he puts the sequence into the computer and it has the computer fold it a million different ways and comes up with the best fits. And one of the best fits looks like this. So this is stem 1, stem 2 is in blue, stem 3 is in green, et cetera, et cetera, et cetera. So this is, we're coming in from this end and going out from that end over there. Very recently, and to give you an idea of how fast this field is moving, the pseudoknot itself was solved by cryo-EM from an interdisciplinary group from many different places, but Ram Das from Stanford is the corresponding author on this. And so this is a cryo-EM structure. This is the real structure at about, I think it was 5.2 angstroms. And it looks a lot like the computationally predicted structure, except interesting, the computationally predicted structure has 2 holes here and here. This is also seen this is a free pseudoknot, so it's seen here and here, these 2 holes, and also this ring-like structure. And they proposed that it's these holes and these rings, these provide hydrophobic or sort of soap-like pockets that small molecules to combine to. So this should be very druggable. Now, interestingly, another group, and this is out of, Nenad Bond's group in Switzerland, have solved the structure of the pseudoknot inside of a ribosome. And so when the pseudoknot gets inside the ribosome, there's a lot of interactions going on, and the pseudoknot is actually being deformed into another structure. You can see that this structure has got some similarities, but you can also see other things, novel elements that are coming out. And the last thing I sort of want to point out from the structure is our work with SARS-CoV showed that this region here, loop 3, has a sequence that enables it to base pair to the loops 3 of another one. And so, again, in this computational simulation, we can see one pseudoknot here, the other one here, and they're base pairing here in this loop 3 region to make what we call a dimer. And we showed previously with SARS-CoV that if you inhibit the ability to form this dimer, basically the virus dies. So we've done some molecular genetics. We make silent coding mutants to basically disrupt this attenuator, the slippery site, and the stems. And just looking at frame shifting, This tells us that slippery site is important. So these are slippery site mutants in 2 different cell lines. If we make silent mutations in the slippery site for SARS or SARS-CoV-2, frame shifting crashes. Similarly, if we knock out stem 1, the ability to form stem 1 or stem 2, there is no frame shifting whatsoever. And if we inhibit or the ability to form stem 3, frame shifting goes down about 2 to 3-fold. So really establishes that these stems and slippery sites are important. This attenuator element, if we add it back in, it decreases frame shifting from about 25 to 15%. And then we put in a mutant and we mutagenize it so it can't refold. And frame shifting is recovered. Same thing with regular SARS. So SARS-CoV frame shifting is druggable. Once we had sort of pseudoknot, this idea that frame shifting is important, this group, Park et al. In Korea, did a drug screen and identified this small molecule, which we'll just call MDTB, which inhibited frame shifting. And in our work, we show that if we add MDTB to cells and then in fact measure frame shifting goes down by approximately half. And then Kylene Ken Hall, in unpublished work, has taken the drug, added it to cells, and then infect them with virus, and virus titers go down approximately 3 orders of magnitude or 1,000-fold. So this really suggests that frame shifting is druggable. So where are we going forward with this? We're working with Mike Woodside, again at Alberta, doing molecular dynamics simulations. And then he does these single molecule force spectroscopy experiments, which are really cool. He takes a couple of styrofoam beads, he attaches the RNA to it, and then he levitates them in laser beams. And then he can pull the 2 apart. And so he can measure the amount of force that's being applied and how much they're being extended or pulled apart. And he can get these beautiful, beautiful curves that inform us a lot about the structural dynamics and the structure of the pseudoknot of the structure. So this is 1 molecule at a time. And then Lois Pollock, we're working with, she's using small and wide angle X-ray scattering to really get an idea of the ensemble. How, if you're looking at a group of molecules, identical molecules, but each has a slightly different structure, how many are represented by that? What fraction is represented by this versus that, et cetera. So the data are starting to come in, and Mike Woodside is seeing basically 3 different conformers, or most conformers that populate the population. And you can see that, so these are these force extension curves. It's really cool. So you pull the pseudoknot apart, and it gets to a point where it's so much tension, it rips apart. The base pairs rip apart. And then what you get is a reduced in force, but it gets longer. And so these are these pulling, and basically from these, he's able to identify, which conformers are present. And then, oh, I thought I had something else there. Nope, that's it. And then Lois has also identified basically 3 different populations of conformers which are present in the Sachs analysis. So this is really telling us how many of these pseudoknots are real, which ones are real, and how many are present at any point in time. And so this is really exciting stuff. And then we're now doing a virology again with Kylene Ken Hall, and she's showing we add MTTB and into the cells, and basically viral titers go down about 3 orders of magnitude. And this is just another curve. And now she's working with humanized ACE2 transgenic mice, showing that they're susceptible. In parallel, we're doing some drug screening, and I'm not allowed to put our data up, but we've identified a number of compounds which appear to inhibit frame shifting. And really importantly, a lot of these are FDA approved already. So we're really pushing forward with that. So with that, I guess I'm finished. Yeah, so that's the end of the talk. I had my acknowledgement slide here before. So with that, yeah, I'm going to stop sharing my screen and I'll start taking questions. So thank you very much.